Multipotent stem cells are referred to as adult stem cells. They're called adult stem cells not because you have to be 21 to have them, but because they're more mature than the pluripotent stem cells. We find these kinds of stem cells in umbilical cord blood, for example. It's a rich source of adult stem cells. And they come from you and me from a variety of sources. We have stem cells in our bone marrow, stem cells in our fat, stem cells in the lining of our nose. They recently discovered stem cells in the pulp of baby teeth. And now here's the real blessing from God. These stem cells that were thought to be only multipotent have been shown to be pluripotent as well. In other words, stem cells from the bone marrow, for example, have been changed into nerve cells and heart muscle cells and a variety of other tissues as well. Now, there's been a lot of research using embryonic stem cells, and there's been research using adult stem cells. So, what do you think? How many diseases have we cured so far using embryonic stem cells? Well, the answer is 0. There has been no success treating human beings with embryonic stem cells. With adult stem cells, however, there's been great success in treating diseases in human beings. At the taping of this DVD, for example, we are now treating 65 different diseases in human beings using adult stem cells. Now, at this point in the presentation, somebody usually asks the question, well, if we're having so much success with adult stem cells and no success at all with embryonic stem cells, why is there this big push for embryonic stem cell research? Well, there are 4 reasons for that. One is that embryonic stem cell research is where this research began. And so scientists get locked into that mode of research. Along with that, there's this axiom in science and other disciplines, publish or perish. Scientists want to be the very first to publish success in this area, and so they're going to continue until they do. And then there's the funding issue. Many states have passed laws allowing for taxpayer funds to be used for embryonic stem cell research, but not for adult stem cell research. And so scientists are going to go and use the research where the funding is available. And then finally, there's what I call the abortion mentality. If we are going to somehow say there's something wrong or something ethically wrong with destroying these tiny little blastocysts at 5 days old, then what are we going to say about destroying a baby at 10 weeks or 20 weeks? And so scientists don't want to deal with those kinds of ethical issues, and so continue with the embryonic stem cell research. As we said, there has been many successes using adult stem cells. Real quickly, here are some examples. A 20-year-old quadriplegic woman received transplanted umbilical cord stem cells to the site of her spinal cord injury, and she's now mobile with a walker. In Germany, stem cells have been used to help repair skull bone damage. Or in Brazil, human patients have been treated with their own stem cells to regenerate heart muscle. And these are just a few examples of diseases we are treating with adult stem cells. Here's a chart that shows the various places in our body where we have stem cells, and then all the other tissues that have come from those stem cells. For example, under bone marrow, you can see that those stem cells have produced virtually all other tissues of the body. So in review, we are not opposed to stem cell research. We are opposed to embryonic stem cell research because that involves the death of a little boy or a little girl, someone created by the hands of God, someone for whom Jesus died. Having looked at the basics of stem cell research, we're now ready to talk about another issue, a related issue, Because you see, if you're going to do embryonic stem cell research, you need a lot of embryos. Good grief, I've been cloned. Researchers want to clone embryos and then use those embryos to obtain their stem cells. So it's important for us to understand the cloning process. In the cloning process, you start with a human egg. And of course, a human egg cell, the nucleus of that cell, only contains 23 chromosomes, half the amount that we need. In cloning, you remove that nucleus from the egg. So now you have an egg that is vacant of genetic material. And then you take a cell, say a skin cell, from the person you want to clone. Now every cell in our body contains a nucleus with the full complement of our genetic code. It's just that in a skin cell, for example, everything is turned off except skin. You remove the nucleus from the skin cell and transfer it into this egg cell. Now you've got an egg cell that has a nucleus that contains 46 chromosomes. If you can somehow stimulate this egg cell, fool it, if you would, into thinking, oh, I've been fertilized, then growth will ensue. And you will have a zygote, a one-celled human being. You can allow further growth, and you will have your blastocyst. And there's that inner cell mass of stem cells. And this little boy or girl that has been cloned can now be killed and those stem cells harvested. Here's another chart that shows the same cloning process. We take a skin cell from this man on your left, take an egg cell from the woman on your right. You remove the genetic material from the egg cell and you put in the genetic material of the skin cell. You have created at that point a cloned human being. You allow it to grow. That embryo can be implanted into a woman's uterus and allowed to grow, resulting in a born human being. That's referred to as reproductive cloning. And the vast majority of people in this country are opposed to reproductive cloning. On the other side of the chart at the bottom, we see what researchers want to do. They want to use those cloned human beings, those cloned blastocysts, in order to obtain their stem cells to help them treat various diseases. This is referred to sometimes as therapeutic cloning. Now, because so many people in this country are opposed to the idea of cloning that results in born babies, the word cloning itself has become negative. And so many times you'll hear politicians and others say that they are opposed to cloning, but they are in favor of somatic cell nuclear transfer. Now, class, if you've been paying attention, you know what somatic cell nuclear transfer is. It's a method of cloning. And so we have people running around saying, I'm opposed to cloning, but I'm in favor of cloning. We need to be very attentive to the language that's being used. Now, this is a very busy slide, and we don't have to look at it all, but I wanted to point out to you how many tries it takes in the cloning process. For example, Dolly the sheep. It took 277 embryos to produce one cloned sheep. And let's blow up this quote. A review of all the world's cloned animals suggests that every one of them is genetically and physically defective. Ian Wilmot, and he's the guy who cloned Dolly, said this: there is abundant evidence that cloning can and does go wrong, and no justification for believing that this will not happen with humans. Another quote from Dr. David Stevens of the Christian Medical and Dental Association. We know from animal cloning that the technical problems and dangers associated with cloning will never produce therapies that these researchers speculate could be applied to human beings. In other words, what they're saying is that because cloning results in defective cells, these stem cells produced by cloning, will probably never be effective in treating human disease.